Okay, so we have wonderful talks today, which are totally different from our comfort zone. We're going to move towards the business side. So we have two wonderful speakers today. We're going to start with Tyler Lalonde, who earned his PhD, who has a PhD. He earned his honors Bachelor of Science in Biochemistry from Laurentian University in 2014, graduating with cum laude. He went on to complete a PhD in organic chemistry and molecular imaging at the University of Western Ontario under the supervision of Dr. Lane Yurt in 2019. So during his doctoral studies, he conducted within the London Regional Cancer Program at Victoria Hospital his research, which focused on developing more specific and sensitive imaging agents to improve the diagnosis of prostate, ovarian, and lung cancers. From 2019 to 2021, Dr. Lalonde served as a postdoc research associate in Dr. Liu's lab at Texas A&M University, where he applied phage display technology to engineer novel inhibitors with broad-spectrum anti-tumor activity. Throughout his research journey, Dr. Lalonde has developed a deep appreciation for the power of imaging and the critical insights it offers into cancer biology, diagnosis, and treatment. So he's going to talk to us on leveraging the cell platform, what we can do for your research. Or your
Thank you very much for the introduction. I appreciate that. So I'll start by sharing my screen, and we'll keep the talk very kind of short and light. Ask questions, stop me, or wait till the end, whatever you feel. So yeah, my background is I'm a synthetic organic chemist. I hope everyone can see the screen. And today I'm really going to talk about SAIL, small animal imaging. So small animal imaging and imaging in general has been a big part of my kind of research career, and developed probes to have more sensitive and early on detection of cancer, but also have a background in radiochemistry and radiotheranostics. So at SAIL, we kind of have the complete package of imaging tools. We have everything from PET-CT, SPECT-CT, 7T MRI, DEXA or dual energy X-ray absorptiometry and X-ray imaging, optical bioluminescence and fluorescence imaging, shortwave infrared, and ultrasound. And we're kind of growing. The kind of four instruments you see there on your screen, DEXA, optical, SWIR, and ultrasound, have just kind of landed here in the last few months for our services. So we'll start off with small animal imaging, and small animal imaging can provide a lot of insights. You're now dealing with, in vivo situations where it's intact biology, and you can get all these wonderful pictures, but also quantitative measurements. So whether we're looking at cardiac function, we can see the beating heart in the MRI there. We're looking at tracer uptake and SPECT imaging, biodistribution, neurometabolism, looking at bone degeneration, lean and fat mass, tumor masses, but we also can image tissues, and you can see a couple examples here. I'll just create a laser pointer so you guys can know where I'm pointing to here. Maybe it didn't come on the screen. Oh, there it is. Yeah, just a bit of a lag. So you can see here, we can actually look at the kidney, bacterial cells, excised bone. So we don't just do in vivo imaging. Up to marmosets is actually the model we can go up to, but we can also look at ex vivo samples. We have an initiative right now working with the clinic, and we can look at clinical samples and clinical biopsies coming from patients with liver tumors via MRI. And we heavily focus on multimodality imaging. So if we take a PET image, we can take an X-ray image, we can take an MRI image, and we can perfectly overlay all of these. So not only do we have the expertise to do the imaging, design the experiments with you, and go through all that consultation process, but we also have the expertise on how to analyze the data and present it in a way that makes sense and is quantifiable for your results. Let's start with DEX imaging. I'm just going to give you a brief snapshot into each one of these techniques and really what it can do. DEX imaging is a really wonderful technique because it can measure body composition and provide you data within 25 seconds. So in a 25-second scan, you're given an X-ray attenuated image, a bone mineral density map, and a lean versus fat mass map. But also within those same 25 seconds, you're given all the quantitative data for this animal. This is an example of a rat image here, but you're given the data sets for lean mass, fat mass, bone mineral density, bone mineral content, total weight, tissue area, bone area, and these can be calculated for any particular region of interest you draw on the animal. I'm going to highlight some papers and some examples that use DEXA and really tie it into a little bit of what the platform CLIP and SIS are doing. So this is an example here. It's a spinal cord fusion model where half of the cohort was an SOST knockout. SOST is seen to cause issues in bone remodeling after injuries like spinal cord fusions. So here we can see the measurements were taken for wild type and SOST knockout, and they looked at the BMD of total body, lumbar, and femur BMD. And in all cases, they can see in the SOST knockout, there's an increase in bone mineral density. But then they compared this to micro CT analysis of these spinal cord regions, where they measured CT volume, CT fusion score, as well as precipitation score. And what we can see is that, again, in the SOST knockouts out to 4 weeks, we see this increase in these measurements, which is correlating to DEXA. So people tend to use DEXA for a quick, fast screening tool. And then you can select a couple animals out of the cohort, post-mortem or in vivo, to go into more detailed analysis on micro CT or CT. So this is a really nice classic example where DEXA is being used in the literature. Ultrasound. So ultrasound is a widely used technique in the clinic and in preclinical imaging. We can see it's a bit laggy here, but this is the video is of a needle-guided syringe going into the myocardium. So you can use ultrasound for techniques like needle-guided injections into the myocardium, into the ovaries, injecting tumors into lung, and you can really clearly see the needle go in. So you know you've had a successful injection, whether that's a tumor cell, a nanoparticle, a drug delivery. So I'm trying to tie it into kind of what may be happening on the CLIP side and what your researchers are doing on a daily basis, but maybe not. And if there's information that you need in imaging techniques, just send me an email and I could share more of that with you. But then it's also good for general anatomy. So I gave a couple examples here looking at the trachea, the salivary glands, the carotid artery, the thyroid. Ultrasound is a very versatile technique, quick and easy, and it provides live data in real time of what's happening inside the animal. Cardiac and neurometabolism. So PET-CT is a really good way to look at cardiac and neurometabolism. So I did a lot of surgical interventions and surgical procedures on mice to induce cardiac infarct, but then also track cardiac infarct and the treatment of this over time. And we can see here in real time the beating heart using FDG PET inside a mouse. And we can actually gain quantitative information about the heart, like end diastolic and cytostolic function of that heart. And then the other image we see here is neurometabolism. I don't know why the video is not playing. It could just be super laggy because these images are high data files. But this would be an example, I wish it would play, of neurometabolism in the brain to look at FDG PET and FDG uptake in the brain. This could be after traumatic brain injury. It could be after a drug induction. It could be after a surgery. So these are some examples for PET CT. This is really cool. This is a big initiative of mine, and it's well underway. I'm meeting with the collaborator this afternoon at 4 o'clock for the next steps. But we're entering the space of radiotheranostics. Radiotheranostics this year alone will be a $15 to $20 billion business in North America. It is using alpha, beta, and Auger electron therapies to treat tumors in a very specific way. So you can use these varying radioisotopes that we see here, like lutetium, like lead-212. This is an initiative we're taking on right now with lead-212. And the really cool part about these isotopes is they're not just gamma emitters. Gamma emitting isotopes is how we can see the tumor and the signal on SPECT imaging that we see in this patient here. But they also release alpha and beta particles. So if you specifically tag an antibody that has a target of interest with lead-212, the antibody will locate to its target of interest, hopefully. That's the main goal. And then it will start to emit alpha particles that can help kill the tumor. But at the same time, you can image on day 1 and all the way up to day 7 because of the long-lived half-life of these isotopes to see the reduction in tumor size. So we are currently working with lead-212. It's fully functional and we have it in our facility. And we're going to be starting the first animal studies in April. We're actually the only center in all of Quebec that has a SPECT CT scanner, which is really, really nice to utilize. MRI. MRI has many, many different kind of applications for soft tissue contrast. One of the major applications that you can do is you can use contrast agents that we see here. Some of these movies aren't playing, but this would be a contrast agent looking at the vasculature inside a mouse brain. So we can look for stuff like bleeds. We can look for abnormal vasculature in the brain by using these types of contrast agents. We can look at stuff like stroke. We can measure stroke volume. We can measure tumor size. We can do blood perfusion in the brain. We do a technique called arterial spin labeling, where we can actually label using a very specific radiofrequency pulse hemoglobin to measure blood perfusion in the brain, the kidneys, and the liver. Optical imaging. I don't know why one image is missing here. That's really strange. There was another image here. I do apologize. And when I send the slides, I'll make sure it's back. But optical imaging is a really nice technique to quickly, again, analyze stuff like tumor volume. Here we see a retinoblastoma in a mouse model, a very, very small tumor, 1/10 of a millimeter in size. But we also now have initiated shortwave infrared imaging within our platform. Shortwave infrared is now in the near 2 window. The near 2 window is very interesting because you can see through the skin in the skull, the vasculature inside a mouse, very non-invasively. This is another initiative we're taking on here at SAIL where we're looking at the vasculature in the hind limb. So we're taking the same animal, it's going to get an MRI, a CT, a DEXA scan, and a SWIR image, all within an hour and a half to assess the hind limb in a neuromuscular degeneration model that we have here in-house. So optical scanning is another very, oh, there, now it pops up. These are M striatum cells M cherry striatum cells expressing inside the brain of a mouse. So we saw signal through the skull. So we then proceeded to euthanize the animal, open the skull, and actually see it within the striatum of the brain. That's my platform, but I always want to give a shout out to the other technology platforms that we have here and we have access to. So we have 8 technology platforms that range from proteomics and molecular analysis, histopathology, biobank, immunophenotyping, molecular imaging, so confocal 2 photon, containment level 3, and bioinformatics. And we have an exciting partnership with a company called Illumix. This is very recent. Illumix is a multi-omics company. It allows us to now tap in to metabolomics, lipidomics, as well as looking at very specific panels like CNS panels via a proteomics ELISA test. So this is one of our partnerships. We have other partnerships across the different platforms, but I wanted to highlight this one because it's tying in nicely what we can offer on the multi-omics side now. I'll leave any questions or we move on to the next speaker and questions at the end. Thank you.
Thank you very much for the wonderful presentation. Any questions for this part? Please feel free to just unmute and ask.
Sure, I have a question actually. Thank you for that wonderful talk. Actually, very informative. I do like the platforms that you have to offer there. Quite impressive too. I was wondering, specifically for the DEXA scans, can you visualize, is it sensitive enough to visualize calcium deposits in tissues such as cartilage and intervertebral disc?
So actually, we just had a discussion. We have a demo unit right now, and we've got the funds to purchase the full unit. And the unit we're deciding to go with can actually do that.
Wow.
So we've decided the demo unit we have right now is really a dedicated DEXA scanner. However, we came across another company that has a DEXA scanner, but also a higher resolution X-ray scanner that can look at calcium deposits. I was just shared some images today from the company. So yes, we will be able to offer that with our new scanner coming in.
That's something that we're interested in, so we might be seeing more of each other soon.
Okay.
And the other question is on the radiotheranostics. So do you have to like vet or select the antibodies of choice? Because I know not all antibodies get internalized by the tumors. So if you're looking at, targeting tumors specifically with antibodies, is this something that you have to look into before you choose the therapy?
Yeah, so that's something you can look into. We don't just need to have the ability to tag an antibody. We can tag small molecules as well,
Okay.
peptides, proteins. So any kind of drug of interest that you're confident gets internalized into the cell, has good targeting, we can make sure that that is labeled for you with lead-212 or lead-203 or another isotope that we feel is best fit. And we could do some initial testing in cells and then move that into in vivo imaging.
Perfect. Thank you.
Yes. So what sort of for the PET, CT, and radio diagnostics, really wonderful, and it's really powerful. So what sort of radio tracers do you have in-house, or do you make, are they custom made or?
So we have a large list of radio tracers in-house, stuff for dopamine receptors, looking at pain, FDG. We have different zirconium-89 tracers looking at fat, changes in fat masses. We have a kind of a big standard list. However, we have the abilities to look at custom.
Okay.
And we can look at a compound that you may have in mind and label that with a custom isotope that we feel is best for the experiment. Because in some cases, if you tell me I want to look at lung function with a nanoparticle over time, zirconium-89 to that nanoparticle tagging is going to be the best option for you to look at that over time.
Right. Okay.
Sorry, to follow up on that, how about nanodiamonds? Can you track nanodiamonds easily?
I've never done that. And I'd have to get back to you on that question. Because I've done all sorts of different like lipid nanoparticles. I've done iron oxide nanoparticle tracking, but not nanodiamond. Are we talking the same like diamond? Like is it just
Yes,
like a
yeah.
okay?
No, it's real.
Okay, okay.
Yeah, they could be functionalized. That's why, like, and labeled or conjugated with all sorts of things, including probably radioisotopes. So, yeah, it'd be
if they can
interesting to
be
know.
conjugated, we can track them.
Yeah, yeah, okay,
As long as they can be conjugated, the tracking part we can handle.
Oh,
sure.
that would, I think we'll talk to you later.
Yeah.
Any other questions? So thank you very much, Ella. So we're gonna move to the startups side of things. So Martin Couillard is the manager of the Clinical Innovation Platform. It's a CLIP,
Yes.
the Health Tech Innovation Hub at the MUHC.
You don't, and by the way, Faxon, you don't need to explain my background. I have a slide about my background, so
Okay, good.
don't waste your time to repeat what I'm gonna do.
Okay.
I'm gonna say.
Okay, good. Wonderful. Makes my life easy.
Thanks, by the way, for inviting me. Laura asked me to present about was a subject like the career opportunities for scientists, and I prepared the beginning of my presentation about that. So I will use my own experience as an example, but I think it could be interesting to share. And of course, I'm going to talk about startups.
Okay.
So do you see my screen right now?
Yes.
Think so, huh? Okay.
Yeah.
So feel free to ask questions, and I will try to see if I can see the chat at the same time. I don't see it. Merde. Sorry about that, but okay, yeah, I see it. All right. Because I wanna see the chat if you have any question, or I will ask you some question during my speech, and I think it's gonna be fun to interact. All right. Here's the 5 topics I'm gonna talk to you about, my background, and then some we're gonna explore some career opportunities with a life science background, I should say. Focusing more on entrepreneurship, startups. I will talk about the ecosystem at McGill, about innovations, and of course some couple of slides on the clip, just to make sure that you know what is the clip. I'm pretty sure most of you have been here or know about, but I have some slides about it. Briefly, my background, I divided in 3 tracks. My training, so a PhD in biochemistry, postdoc in human genetics, and then also I try to orient my career towards technology transfer, kind of, bringing an invention, I should say, from the university to the market. So there's a lot of steps, a lot of things to think about. So there was a diploma at that time. It was back in 2010, so it's been a while. It's no longer exists. So there's no training for people that want to become a tech transfer manager, you know what I mean? So it's a very niche background, a very niche experience, expertise it's hard to get. And by that time, I also see in my work experience, I put some stars around startups, because I tried to start some startups while I was at the postdoc. I was trying to find a job. And briefly, it was not successful because of many things. When you try to think about a startup, when you have a team of only scientists, there's no business people in that, so the project doesn't go very far. Or if you have some good teamwork, maybe the project is not so solid. So many things that can block an idea of a startup. But back in that days, there were no ecosystem, or there was no culture first, and a strong ecosystem to help to launch a startup from the university. Now it's different. That's what I'm going to show you today. So talking about work experience, so I tried to find a job in the industry, and I had the chance to work at Roche Diagnostics, so the site of the diagnostic here in Laval. But it was very there's no R&D going on at Roche. It's in Canada. It's more the R&D is done in Germany, Switzerland, and in the States. So I felt I was not so stimulated. My scientific side was not so stimulated, and I want to focus more, as I said, the tech transfer idea. So I had a chance to work in an IP law firm to know more about patents, and then had a switch to more the research office side of tech transfer, or we can say valorization. I will talk to you about this term later. At the HEC Montréal and also at the Western University. I start a new office at Western, but based in Montreal. I was scouting technology startups from Quebec with a specific program of the tech transfer office of Western to bring some startups at the international level, especially in Asia, because we had employees in China, Japan. It was crazy. And I had the opportunity to join the launch of the new entity for tech transfer activities in Quebec called Axelis. It was back in 2021. They launched their activity down there back then. And then at that time, I was able to start my EMBA because it was encouraged by my employer. And then I had the chance to start recently last year as a managing startups, so it's a little bit more downstream of the invention, something that is more tangible, I think, which is more impact. We are closer to startups. And all the being said, the last track is the volunteering. I mean, it was more focusing on networking. That's a take-home message that I can give you today. You have to think about how to interact with and to. You don't do networking necessarily in the way to find a job, but to develop your network in the field in the field of expertise or in the industry that you would like to work on. In my case, at that time there's Biotech Anecdote. It was more for life science scientists like you and I, and a mix of industry and academic. So it was fun. I don't think they're very active now. The Licensing Executive Society is something that is more focused for licensing for more, not lawyers, but, negotiating terms for licensing technologies. I was the chair of the Montreal chapter. So the idea is to create events about specific topics. So when you are an organizer of events, it's always fun to interact, and you can talk to people that normally it's hard to talk to. Same thing for the FORPIC, which is the Forum of IP, Quebec. The Forum de Propriété Intellectuelle. So I can give you an example. In 2020, 2021, I organized an event where I knew that the Axelis was about to be launched. So I contact the people way up from the launching of this entity. I say, hey, would you like to do a conference, or at least a panel about the tech transfer in Quebec at the FORPIC? And the timing was so great that by doing so, I was able to talk to the manager of the future entity of that's going to be Axelis, and they hired me because I was able to develop a good networking. So being involved in this kind of activities, it's always a good way to network. So last thing about networking, it's very important. I think when you meet people in these events, try to send them a LinkedIn invite, say thanks, or a small word, say it was nice meeting you. And you never know, you're going to repeat, you're going to see the people very often in these kind of events. The ecosystem is very small in Montreal, so trust me. And if you're really serious about networking, you should network with someone every week. You should have at least a coffee with someone new to build your network. You have to be very active. So, example of career opportunity. That's a slide that has been created 10 years ago. So when I was graduating, and here's some career opportunity focusing on at what level, at what part of a cycle you're going to focus, because you cannot do all the cycle of creating a product. You have to be focusing on more design, the prototype, the development, or the marketing of it. So when you work in an industry, you cannot do the job of another person. You have to focus on a specific project, a specific expertise. And as you said in my previous slide, I did some of that, and by that time, the entrepreneurship and startup was not in the pipeline really when I was thinking about career opportunities. That's what I'm going to talk to you about today. So, and I highlighted some expertise. I gained some experience during my career. So we can take some time if you want to discuss. If you come to the clip, come to see me. We can discuss. Or if you have some questions during the webinar or after the webinar, I can answer some of your questions. But there's some pros and cons about every positions. Okay, let's dive to the startup words. Let's start by a question about a question about feelings. Do you feel at McGill or in your department that you have there's a culture of innovation here? You can type yes, no, or put your thumbs up. I want to just feel the audience. Do you feel that at McGill, innovation it's not only doing a research in publication, but when I say innovation, it's okay. I will have an impact. My study will have an impact in the society. Not necessarily commercially, but you know what I mean. So and even Tyler say no. Okay, good. You want to say why you say no, Tyler?
I just feel maybe not a hard no. I think there's a lot of growth that we need to do to continue this.
There's a lot of work to do.
I've only been here 7 months, and since I've been here, it's been good. I've started 5 new partnerships to bring into the platform. But I feel there has to be a broader connection across all of Montreal and Quebec for us to continue to grow this as a team.
Okay.
And I think it could be really useful on how we could grow this as a team. So I think that's why I said no. I think we're getting there, but there's some work in the background.
And for sure, and a way to increase the innovation is to answer the second question: what are the entities that are existing at McGill because we need to work to collaborate more. There's a lot of silos that you may hear. Is there some resources that you know about at McGill? And when you're here, you think about innovation. What will be your reflex to? Is there? You want to type in the chat some ideas? There is one that should be obvious because it's in my name at the end of my name. Yes. No ones? Do you know, of course, the clip, but do you know about McGill Engine? Yes. Is there another incubator in McGill? Don't have any idea? Have you ever heard about Dobson? It's another incubator for McGill when you can learn your business abilities. And of course, you need to know also the innovation and partnership office, what we call the technology transfer office. I will add some note about that in my next slides, because it's where if you invent something, you can have some help from them. And do you know a spinoff or a startup from McGill? Because there is some. No one? Yeah. Yes. Great. Taiga, I can check. Of course, I don't know all the startups because there's a couple, and some of them are in engineering. So it's not my field. I will show you some logos. Neuromedical, yeah, that's a good example. And they were in the clip. So let's see. Here's last year there was a paper about saying that McGill is one of the in North America the best university to create startups. Good to know. So so talking about the culture or you are now more surrounded by these kind of initiative of startups. See the ranking on the right, number 2 in Canada. So that's good. I think UBC is the first, and 23 overall in the world. That's in terms of number of startups. It's cool. Let's see some logos here. In healthcare, we have maybe you can recognize some logos if you're more familiar with startups. And then also in engineering, we have these kind of startups. That Sportlogics, it's something that it's at the bottom right here. It's very famous to the stats, to analyze stats of the Montreal Canadiens game in the NHL, for example. Some stats that I think is interesting to know. We said that there's a lot of startups coming out of McGill. I see the red arrow is pointing that 10 years ago we had only 1 startup, and now we have 28. So it's a big improvement. And it all starts from an invention disclosure or report of invention that they receive at the Technology Transfer Office. So also an increase. And interestingly, most of them we try to patent. Back in 10 years ago, I was talking to the manager of the director of this office, and they say we were about trying to file patent on every invention disclosure. So there was a lot. So they work hard to focus more on quality rather than quantity, and now you see that there's less patent application. So, but which is a good sign, I think, because they're focusing on the quality. At DRI, do you have how many invention disclosure that we get in 2023? Do you think we at DRI we are strong, or more than 50% of the declarations? Not sure, huh? So I got the number. It's at about only 14, so 11%. It's not a lot. So that's why we have a lot of work to do for the culture. And I'm pretty sure in your lab there's a couple of ideas that might become an invention disclosure in the upcoming months or years. But you need to know that that there's room to improvement here, and you need to know, if you think about that at McGill, they have IP policy, and there's some guidelines that you can read about if you want to know more. That we have a joint IP policy at McGill. Maybe James Mehta talked to you about that in his previous talk. I think he had a webinar in October, November. But I just want to repeat that. It's important to know that McGill is very a policy that helps you to develop your ideas. If you want to do it with McGill, they're gonna help you, and they're gonna take a small portion of your of the revenues. We can discuss that in more detail if you want later. But there is also the possibility to do it by your own. But if you do that, you don't have access to all the services from the McGill. Because McGill can pay for your patent, you can have access to grants. The McGill Innovation Fund, or so they provide money to the startups. So it's good to know. Take home message: there's 2 possibilities that you can think about when you try to you want to launch a startup. Because when you want to start a startup, you're gonna get a license from McGill. What you create at McGill, there's IP belongs to a McGill, so they're gonna give you a license to allow you to do a startup. And by the way, in the state, it doesn't exist. There's in the law, there's a Beta Act saying that all the IP that are generated in the university belongs to the university. So see, in Canada, we have a mixture of IP policies across all the universities. Some that are inventor owned, some of are only university owned, and at McGill we are joint IP policy. It's interesting to know. I think I was not aware of that when I was a student. So let's take a step back just to make sure that I should have started with that. I wanted to talk about valorization, and innovation. So the term valorization is very a French term. So valorisation. So, but there's no counterpart. It's hard to find a word that is covering all these expertise at the same place. So I'm assuming everybody here is doing some level of valorization of their research. Mostly if you're doing collaborative research but you're transferring knowledge to you're creating knowledge and you in exchange with a community. It could be scientific community. So in our case, today we're talking about the second one, the entrepreneurship, but it's also connected to the last one, commercialization, because when you invent something, you can license it to a big pharma or a company. So it's a license. So it's going to be this last part, but you can also get a license from the university, as I said. So you're going to do a startup, a spin-off of McGill. So it's entrepreneurship. And what is innovation? So you can imagine, since I'm talking, you have a better idea, knowledge about that. So it's at the level, and inventions at the level idea. Innovation is more the application out of it. So that's why we say a scientist seeks understanding, an inventor seeks a solution, and an innovator seeks an application. And this last part, it's a long process. There's a lot of things to do. So that's why when you invent something, sometimes they say, it's worth $1 million, so I will become rich. But if you want to become rich, you need to do the last step also. You need to be involved in the last step. So how to create a good innovation? These days, it's the magic occurs at the intersection of the many expertise, like clinical engineering and business, and all the steps at the right that you need to think about to create, to bring your invention to an innovation. And that's why at the clip there is in the department, the program of the SIS, there's a surgical innovation program that is based on this idea of mixing, business people, clinicians, engineers, you know what I mean? To create, to focus on a real problem, developing a solution, focusing on starting from a real need, an unmet need, and you can start a couple of there's some good success story like neuromedical that has been mentioned in the chat. It's an example of this program, the success of this program. So the clip also has been designed has been created based on that. So you're trying to find a place where the business people can meet the clinicians, and we can and the academics to create the projects and eventually help to develop to bring the startups to grow the startups even bigger and eventually get an exit, let's say get acquired or even launch product on the market. So as you may know, it's based the clip is based at the Montreal General Hospital. We're on the 9th floor in the C9. It has been recently renovated. So you see a picture of that has been taken, I think, before COVID or just after. It's empty a little bit of the walls, but it's just to give you an idea of what it's looked like if you have never been here. And I will say that the services are more in 3 tracks. The first one is the great added value to the ecosystem of incubators or for the startups. Because if you go to Dobson or if you go to Santec in downtown Montreal, this part is an added value. So the clip is collaborating with all the incubators to help a connection with the MUHE, the clinicians, to validate to do some clinical validation. And talking to what we call the KOL, the key opinion leaders. It's very important when you have a startup. And then, of course, the last 2 columns, it's more the infrastructure that's to help this ecosystem, having access to labs, 3D printers, some help to do some clinical study and to do your REB application also. Access to office spaces. We have a big conference. We're doing events that you can attend. here's a list of some KOLs that specialties that they are the expertise of the KOLs that are connected to the clip. Some partners from the network that you might know, the SIM, the animal facilities that Tyler presented is all the imagery and all what you're doing it can be accessible as a clip member. You can have access to the platform of the RI. Here's some pictures from saying that we do demo day conferences, IP clinic workshops, and some pictures with in the middle. This is we had the visit of Luc Sirois last in May last summer. The Fitzgibbon, where we are. So I'm showing you some pictures of some labs that we have. We have dry labs, wet labs. So and here is to give you an idea of the number of startups that we have connected with some KOLs, and also we have corporate members that we try to grow. For example, Johnson & Johnson, we have Stryker. They're doing projects with some clinicians at the MUHE. And of course, we have, by being a member of the club, you can have access to some funding opportunities, like the Montreal General Hospital Foundation Impact Investment Fund. So they can invest in the startups, and there's 2 calls per year. So the next one is going to be in June. So it's very interesting. And if you apply, your project will be evaluated by a committee, a funding oversight committee that is an independent expert in the field. So it's good, and you're going to get some feedback from these experts. And there's also the clinical innovation competition. The deadline is now, March 31st. And the startups can win prizes. And there's a new RAB Student Innovation Prize this year. It's new. It's the first time that we're providing a prize for students. And they're going to be an event in May 21st. So I would like to finish with this slide about the ecosystem. It's going to maybe help you to understand a little bit better where the clip fits inside the McGill ecosystem and the McGill and the MUHE. So if you start on the left with, for example, the program, the surgical innovation program, or you create something in your lab or at McGill, or all the other incubators like Engine that I talked about, Neurosphere, Dobson, and the MIF, it's a McGill Innovation Fund. So the startups are receiving money to develop their technology. They can, as a startup, enter the cycle of having an invention and trying to do some validation. But with their prototype, they can test it inside the simulated environment like provided by the clip. And there's the Steinberg Simulation Center here also, all the partners that I've talked to you about in the previous slide. That's a point of entry to the MUHE. If you want to eventually do clinical evaluation or to talk with KOLs, you can get some feedback out of it. So that's why the clip is here as a point of entry for the clinical part. So the more you go on the right side, the patient safety and the risk is, you can reduce the risk of your technology. So going towards from the clinical need to a clinical solutions to develop your product. So we have many other partners, but I want to show you this slide is trying to be simple, but I think it can give you a better idea. So recently at the MUHE, they created the Impact Center. They call it it's a joint innovation council that's gonna help to prioritize and help to identify some problem in the hospital setting that we can nurture the cycles of creating new innovations out of it. So there's a good timing in the ecosystem that to launch a startup based on particular needs. So I'm finishing on that. So of course, as a CEO or have a startup, you have to think about a lot of things, but there's an ecosystem to help you. The clip is part of it. So I'll be available to talk when you have some ideas or and so on. So you can have my email address here, and you can send me an invite on LinkedIn also. Let's connect, and I'm open to if you have any questions about startups and entrepreneurship. You can ask question right now or come to see me afterwards. Thank you.
Thank you, Martin. Questions? Anybody?
Yeah, thanks for the talk, Martin. Much appreciated. I love how this is where I want to try to bring our ecosystems together, what you guys have going on here, and what I'm trying to build here is kind of that similar expanded ecosystem, but then offering that into everything we have across these different platforms. So just to kind of expand a little bit and not too much, I was in Boston last week in Kendall Square. I met with 9 pharmaceutical companies in their offices with their directors.
Yeah.
We're expanding projects and we're going to bring them in as external clients to run projects through our platforms, but then inviting them into our total ecosystem with other services we can offer at McGill. And that's what I'm trying to do, because the more we can offer a client that I'm speaking to, the more we can retain that client for multiple projects.
I think so, yeah.
So yeah, it's good to know that I can hopefully try to introduce my clientele basis to everything that's happening in the back end of McGill and all the other hospitals and everything kind of going on in Montreal.
Yeah, and the clip, it's not in the back end, it's the front end.
Yeah, sorry.
No, we brainstorming, but and I have a couple of good discussion with Benoit Chambaron also at the DRI, the research office, about this kind of how we can help to convince a company like that, a big pharma. If every silos are talking to the same partners, but what about showing that at McGill, or at least at the RI first, there's a lot of services and expertise that can be helpful for them as a partner.
Yeah.
So that's why I think they can work on being more and more able to sell ourselves with all this expertise.
Yeah, and that's exactly, some of my partnerships are solely going to be, we get to utilize their instrumentation and tap into that instrumentation that we don't have here, which is really neat. And some cutting edge instrumentation that we just may not have the funds to buy now, but we can tap into to help anybody if they have a company growing and they want to do neuro-specific panel of proteomics. Well, we have an instrument that can do that now through our partnership. So yeah, very, very nice talk. Thank you.
Yeah, thanks. I know, a very important topic. Michael, do you have a question?
Yeah, sure. I'm really happy to hear that. A wonderful talk, by the way, and I'm happy to know that McGill is doing very well as an incubator for new startups. I guess my question is related more to the current situation, with our neighbors down south and our wonderful president down there, and the whole economic scenario that's going on. Will that affect like funding for such things, or do you foresee it to affect anything in the near future?
It could affect the fact that as a startup, the first market is the United States in medical, and you develop a medical device. You cannot just focus on Quebec and Canada. The market is not big enough. So of course, if the relationship is not so good with the United States, it's going to affect what was more smooth before Trump, and maybe the VCs, because we need in this field, you need money from venture capitals, VCs, and most of them are American. But with this level of uncertainty, there's more risk, so they will wait. So what we're going to see is less investment just to see if the market will be open for investment because we need the US market in some way, depending on the device that you develop, for sure. But most of the time, the US market is one of the most important that you have to focus and you need to enter. So if the investors are waiting, it's going to affect the cycle. It could be the opposite. Okay, no, let's see, but maybe I would like to invest more in Canadian companies because I don't want to invest in America no more. But for now, I see more step back for now and a wait and see situation, which is not good.
Any government incentives or initiatives for that to maybe help maintain these startups?
But
You see that happening?
I think we're going to see some initiative like that, but they are not so agile. So by the time they create a new program to help, for sure, all the conferences that I'm attending, and the one yesterday at the Glen, they called Research Canada was the MUC yesterday. They were saying that the people from Research Canada are trying to find ways to help the research in Canada to distinguish yourself from what's going on in the US. Same thing for startups and innovation. So let's see. I don't have I'm not aware of a specific program for now.
Yeah, and when I was it was sad, but I guess a blessing for Canada. When I was in Boston last week, I was entering pharmaceutical companies, and these are billion-dollar companies, where we would walk into a room, they had $40 million in infrastructure in terms of instrumentation and no one to run it. Wow. So layoffs are happening, but the good news is they said to us, well, we need you guys now because you have the identical infrastructure, but you have the expertise, knowledge, and people to run it. And they're outsourcing that. And this new partnership I started, the company's out of Sherbrooke, but they sell their research hours in terms of blocks of hours. So last year, they sold almost 10,000 hours in research hours, of which half are still to be used by their clients this year. It's a unique model because it's paid up front, research hours in dollars. And the cool part is they're going to allow us to kind of insert ourselves into that mechanism by offering what we can do here through their already paid research hours, which will then lead them to top up more and buy more. But yeah, we noticed that a lot. Like I said, I attended 2 events, networking events through BVS, which is biotech vendor services, and attending all of these different pharmaceutical companies. That was the message was, we have the equipment, but not the personnel to run it. So, which was interesting.
Yeah.
So any other questions, comments? So thank you very much, Ilan. Thank you very much, Martin. And thank you everyone for attending.
Faxon, we have a student to present. Yeah, we have a student.
I'll go to sleep. Oh, thank you. Okay, let's move to the student.
Okay. Suman, I think she was supposed to moderate. Suman, are you here?
Yes, I'm here.
Okay, perfect.
All right, good afternoon, everyone, and welcome to today's student seminar. My name is Suman, and I'll be your moderator for this session. It's my pleasure to introduce our student presenter, Georgi Danilenko. Georgi completed his bachelor's degree in electrical engineering at McGill University. He has a strong passion for hardware design for new medical devices, which has driven his current research. Under the supervision of Dr. Thomas Hemmerling, Georgi will be presenting his project titled Assisted Video Laryngoscopy, a novel method of endotracheal intubation using a next-generation video laryngoscope with artificial intelligence assistance.
Thank you, Suman. And thank you, Dr. Lalonde and Martin, for presenting today. Yes, my name is Georgi Danilenko. Is everybody seeing my presentation in the correct screen? Do you see
Have
my
a
presentation?
very small slide.
Yeah?
Yes.
Okay, okay. Let me try something else. 1 second, 1 second. 1 second.
Now we have a big slide.
Yes, yes, 1 second.
Okay.
That should work.
Okay,
Yeah.
is this good?
Yes.
Good.
Okay. So, my name is Georgi Danilenko, and today I'll be presenting to you assisted video laryngoscopy, which is a novel technique for intratracheal intubation using a next generation video laryngoscope. So I have no financial conflicts of interest, and this presentation is purely educational, and I recommend no procedure in this presentation. So, did you know that when you're being put under general anesthesia for surgery, you might not be breathing for up to a minute? Well, cerebral hypoxia can take less than 5 minutes to cause brain damage and death. So, what is being done to improve and speed up intubation? What research is being done? Well, in the US alone, 400,000 emergency intubations are being performed annually, and 12.7% of them are unsuccessful on the first attempt. A bad rate of first pass success rates and a slow time to intubation can cause complications, such as oropharyngeal trauma, oxygen desaturation, esophageal intubation, aspiration, and other complications. Currently, there exist two techniques to intubate patients under anesthesia. The first one is direct laryngoscopy, which utilizes a direct laryngoscope, such as the Macintosh, to visualize the glottis directly and insert a breathing tube. The other technique is a newer technique that uses a video laryngoscope with a screen and camera to visualize the glottis indirectly and insert a breathing tube. However, as of now, there's a lack of evidence in academic literature to advocate that video laryngoscopy can fully replace direct laryngoscopy. This is significant because medical professionals and hospitals are forced to train in both techniques and buy both devices. Furthermore, literature shows that the first pass success rates and time to intubation are suboptimal, and this can cause complications in patients. So the hypothesis of my research is that a novel assisted video laryngoscopy, short AVL technique, with modern technologies such as AI-powered visual guidance, can improve first pass success rates and time to intubation, reducing complications. So AVL, as I will call it, it may fill the gap in the lack of training and assistance through augmented reality on the screen of the device. So this could be such as indicators of the optimal blade tip positioning, indication of the blade insertion distance, as well as a bounding box for visualizing the glottis and a green light to tell you when you're good and well aligned to intubate. AI models like the YOLO V4, they may minimize the negative effect of difficult intubation. Furthermore, there has been a recent research that showed that there's a 14.3% improvement of first pass success rates of video laryngoscopes compared to direct laryngoscopy. And considering that assisted video laryngoscopy is similar to video laryngoscopy, but has the added benefit potentially of AI, then the hypothesis remains that it's possible that AVL can be better than both previous techniques, and especially for personnel in emergency situations, which are either can be under a lot of stress or have less experience than anesthesiologists. Therefore, the objectives of my research is first to perform a survey of video laryngoscopy and direct laryngoscopy preferences and uses by anesthesiologists across Canada. Then it's to develop and produce a assisted video laryngoscopy prototype with hardware that is required to run an AI model. And finally, to trial this technique against video laryngoscopy on an airway mannequin at the MGH. So first, the survey was a 20-question electronic survey sent to anesthesia practitioners across Canada, so trying to reach 3,393 of them. And the survey was divided into anonymous demographics, the availability of video laryngoscopy, the preferences and competences of video laryngoscopy and direct laryngoscopy, along with some other questions. The prototypes were designed using a standard research and development engineering procedure. So a chip was selected, an NVIDIA chip, to run the AI. A carrier board for that chip was designed with Altium and manufactured. A device body was designed with Fusion and also 3D printed. And this would be all integrated into a version 1 prototype that has a screen attached via cable. It is handheld and has an off-the-shelf carrier board. The purpose of the V1 is to build the AI model on which we will intubate in the trial. The second version, V2, has a custom carrier board that is smaller in size and that works as a proof of concept that we can indeed put AI on a video laryngoscope. Finally, the mannequin trial involves 20 participants with no medical experience. 10 participants would do 10 intubations with the traditional video laryngoscopy, and 10 others would do 10 intubations with assisted video laryngoscopy. This is all filmed, and measures would be extracted from the videos, such as first pass success rate, time to intubation, time to best Kermack-Lehane grades, and Kermack-Lehane grades at intubation. The survey had a 10% response rate, and it showed that participants, anesthesiologists in Canada, 83% of them prefer direct laryngoscopy for routine intubations, whereas 71% of them switch to video laryngoscopy when they encounter difficult intubations. Video laryngoscopes are also overall very accessible across suits in Canada. And the COVID-19 pandemic prompted a 28% increase in video laryngoscopy usage. However, the study also showed that there's barriers to video laryngoscopy, which include anesthesiologists having a greater skill in direct laryngoscopy, and the cost, maintenance, and environmental concerns related to video laryngoscopy. My findings support literature. So direct laryngoscopy is indeed suggested when it's a simpler, more routine intubation, and video laryngoscopy is suggested when it's difficult. However, this comes back to the reasoning of my study. You have to train on both techniques and have both devices. So can assisted video laryngoscopy be the replacement for both? This is the version 1 prototype. It has been built and it feels and performs just like a typical video laryngoscope device. It has a off-the-shelf carrier board located behind the screen due to size constraints. And the AI was trained with this prototype. Next, the version 2 prototype has a custom carrier board designed, and it features 110 millimeters by 52 millimeters of size. And it was used for the mannequin trial, and it's a proof of concept that we can put AI integrated into a beautiful video laryngoscope. Finally, the mannequin trial is currently underway. 20 videos were recorded, and then measures would be extracted, and data will be analyzed, and conclusions will be made. So let's recap. First, a survey was done to verify if literature is correct. So yes, video laryngoscopy and direct laryngoscopy have their use cases, but that means that we need to have both techniques and buy both devices, and there's a lot of devices to buy. And there's barriers to video laryngoscopy, even though it's a newer technique. So the question is, can assisted video laryngoscopy trump both techniques, beat the both techniques? Secondly, the prototypes are working, and the custom carrier board is a proof of concept to pave the way for integrating AI in the video laryngoscope, as well as other technologies. And finally, the mannequin trial may show advantages of assisted video laryngoscopy over video laryngoscopy. So will the participants have a faster rate of learning or a better visualization technique with the help of AI? I think this research is important and has strengths in the sense that the new device could be easily integrated into current hospitals and given in the hands of practitioners because it is similar to a video laryngoscope device that we have right now. And it is developed during the peak of AI and other technologies. So there's a lot of work that can be left to improve this, done to improve this. However, my research specifically has a weakness in the sense that the mannequin trial is small in size and there's many variables that can affect and give imperfect results. And there's many questions left unaddressed, such as the concerns of the practitioners. To finish up, I would propose additional work on this research, and that would be creating a trial study instead of mannequins on cadavers to have a more realistic approach to this, and using a version 3 designed assisted videoendoscopy device similar to what to the quality of what is on the market. Finally, I would suggest adding more technologies into assisted video laryngoscopy, such as better AI models, as well as maybe robotics. And the devices should address cost, maintenance, and environmental concerns of the practitioners across Canada. So thank you for listening. I would like to acknowledge and thank the Department of Surgical and Interventional Sciences for making my research possible, as well as my research committee, advisory committee, and the ITAG laboratory of McGill, the partner company, Devoco AI, as well as the funding organism, the MITAX. I'll be happy to take any questions you have.
Thank you, Georgie. Wonderful presentation. So you have 3 questions in the chat. One from Fatma.
Yeah. Okay, let me
Can
see.
KVR be used effectively across all age groups, including children and elderly patients with different airway anatomy?
Yes, that's a good question. This would depend on the blade size. So it's a purely mechanical question. AVL is focused currently on AI assistance, so visually. It is possible that having child or elderly patients or even trauma patients would differ the anatomy, and AI models could be trained to help assist the anesthesiologist in finding the correct positioning of his blade. And also, yes, you would have to essentially train multiple AI models to accommodate every population. So that's a very good question. So, okay, are the anesthesiologists aware that it will be difficult in advance or that they switch to the video after they failed? Okay, very good question by Marianne. From what I read on literature, most of the time they are aware already because of their experience that it's going to be a difficult intubation. The most obvious case would be when there's trauma. However, in my survey, I do ask what happens if you encounter a difficult intubation, and I have the word switch. So it is possible that they try first time with direct and then switch to video laryngoscopy if it doesn't work out. Then again, failed first pass success rates are only at 12.7%, according to what I presented here. So it doesn't happen very often. They would either choose direct laryngoscopy initially or video laryngoscopy initially. Okay, any results of assisted video laryngoscopy system? First pass success rates. Okay, yeah. There are results, but the mannequin study essentially is still underway. It's still being analyzed. I couldn't answer you without having any bias. Yeah, so no results for now. It's still underway. Okay, from Muskan. Is AI integrated directly in the laryngoscope device, or does it work via an external system? Yeah, good question. So that's the whole point of the version 2 prototype. The version 1 cannot have AI directly integrated into the laryngoscope device because the off-the-shelf solutions are too big to accommodate for a handheld device. In fact, the version 1 is handheld, then you have a cable going to a screen, And in that screen box, you have the AI system module. The version 2 is a carrier board that is smaller in size that can run AI. And the whole purpose of the version 2 is that, yes, indeed, you can make a handheld device with AI in the handheld device with a screen on the device. Okay, thank you. Yes, thank you as well. Another question. Does this device incorporate any real-time feedback mechanism? And during intubation, and were certain engineering challenges like latency, resolution, and elimination? Okay, so this would be part of the drawbacks of my research, as I don't have the full capabilities to design something as quality as something that is on the market. So absolutely, you would, if you were to go forward with something like this, you would have to tackle latency, resolution, uh, the your display, your camera, your illumination, everything. You would have to tackle it. So there, it's making essentially almost like a computer in your hand. And there is no current real-time feedback mechanism in my prototypes. However, you could always think of maybe adding some feedback like sensors or robotics. Okay, thank you. Thank you. Okay. I think if anybody has other questions, I'm happy to answer or comments.
Thank you very much, Georgie. And thank you everyone for all those very interesting questions. Any other question? So if there are no more questions, again, thank you everyone. And have a wonderful week. Thank you. Bye.